I seem to still have a steady stream of people trickling in, so we might keep waiting for a little bit. Yazan, are you able to share the welcome slide for today? If not, I can. Oh, you're already on it. Too good. Okay, well, we might get started at least with the introductions for today. Um, So, welcome to today's Biocurator Workgroup meeting. The theme for today is going to be all things gene curation within ClinGen. And so we're going to have Courtney Thaxton and Marina DiStefano on behalf of the Gene Curation Workgroup, who will be leading us through a sort of Q&A style session. So we had a survey that we sent out at the beginning of this year that I know a lot of people submitted questions to, and those questions will all definitely be answered today. I'm going to go off script here, but I'm assuming that if anyone has questions that arise throughout the meeting, that we're happy to be taking those questions too. So either put them in the chat or raise your hand and we can address them live. But if there's not anything burning before we get started, then I will hand over to Courtney and Marina.
Great, thanks, Alicia. So I will start by sharing and then oh, wow, I'm not a host here. I have requested to be. Okay, wild day for me. Okay. So thank you to those who have submitted questions for this. So today, as Alicia said, we'll talk about question and answers. And so the agenda for today is we're actually going to review some recently released functionality in the Gene Curation Interface. I think we had previously gone over these quickly, but we just want to reiterate when we're talking about question and answers. So these will include scoring ClinVar variants, autosomal recessive phase status, and editing published evidence summaries. We will then also work on answers to questions received ahead of time from the survey. And then if we have time at the end, we will also have a gene curation question and answer. And just to set some guardrails on that, we'll take questions about curation only. If you have any other questions related to ClinGen at this time, please do email the general ClinGen email address here, clingen@clinicalgenome.org with those questions. But we'll be happy if we have time to take questions about gene curation. Okay, so let's talk about scoring of ClinVar submissions for gene curation. So this was a policy decision within the Gene Curation Working Group that on a GCEP by GCEP basis, of course you want to check in with your GCEP, that GCEPs could score because they are publicly available ClinVar entries. However, I feel obligated to say the required caveats for this. You should feel that your GCEP should feel that they have enough phenotype information to score a ClinVar entry. Oftentimes, we have internal lab members from clinical labs on an expert panel, so they will have additional phenotype information. So it's difficult to just on the surface score a ClinVar entry unless it's like a Genome Connect entry that has a lot of phenotype information. So that's one caveat. The other caveat is if you have an autosomal recessive gene curation, you really need to be checking in either with that submitter or internal lab member, et cetera, because ClinVar entries are assertions at the variant level. They're not case level entries. And so you're really only seeing a variant at the ClinVar level. So you need to make sure that there's a biallelic case observation if you're scoring in autosomal recessive cases. So those are just the caveats that I wanted to say ahead of time. And GCEPs are not required to score ClinVar entries. This was really a request based on the input from the community that oftentimes if a gene disease relationship isn't extremely novel anymore, that manuscripts or journals will not publish those cases, or people don't have time to publish those cases. And so given the right amount of information, you may score them in your gene curations. So let's talk about how you enter them to the GCI, because previously you just put them in the evidence summary text and add that information there, but now you can capture that granularly. So to add a ClinVar submission as evidence in the GCI, I see there's, oh, and Courtney stuck more info there. So you have now a select evidence source. So this used to just be PubMed IDs that you add. But when you hit the add button now, a new pop-up will appear to enter either a PubMed ID or a VCV ID here. And so you have to include the version number of this. So first you'd hit add, then you include this VCV number, which is the version of the variation here. And then below that, so this is just points to the variant in general, just the variation record at the very top level. However, you also need to further add the provenance of basically who you're getting the information from. And so you need to go further and add the submitter specific ID. So that's the SCV ID, including the version in the bottom field here. And so you can use the VCV link to find the correct SCV link that you're looking for. So that at least will link out and you can go and find it. And that will give you the submitter specific here. And so once you add in the SCV, it will populate. You click the retrieve evidence, which is the next option, and then it will populate the submitter and the variant and a little bit of the information and a link out to the SCV to make sure that you're choosing the correct one. And then you would click the add evidence source here to add this variant. And so just to give a little bit additional information, what's the ClinVar VCV? As we said, this is the accession ID of the aggregated data from all the submitted records for a specific variant. So it's at the variant level. And the number after the period is the version of that particular variant accession. So for example, in this one, it's version 3 because it's .3. And where do you find this? It can be found in the variant details section here, boxed nicely, of ClinVar's variants ClinVar accession subsection and identifiers here. So variant details, this opens up, and then identifiers, and then accession here, VCV. This is usually next to the ClinVar variation ID, which is probably the one that you're a bit more familiar with because this is used for the allele registry or sometimes in the variant curation interface if you're adding variants. So you might be more familiar with the variation ID here. And so you need to add that first, as we said, and then there are the submitter specific accession IDs. And so those would be the SCVs. So this is assigned to a submitter record in ClinVar. So if a lab submits an assertion, they're going to get an SCV. And so this also needs to have the version number. So in this example here, this is version 4. And just because of the way that ClinVar data are set up, we need to include first the variant VCV and then the submitter SCV when we're pulling this to display. And then now the ClinVar submission is viewable and scoreable in the GCI. You can see the comments from the ClinVar submission where you would see usually the abstract for a PubMed ID. And so then at this point, evidence can now be added and scored as usual. So you could see if you wanted to include it as non-scoreable evidence. We would probably discourage from clicking this as the earliest report of a variant in the gene because you would want to use a published article for that. But you can add all of the usual information that you would add for a PubMed ID. In most cases, you'd probably be adding something of the genetic evidence here. So that is entering ClinVar variants there. So I will head over to entering phase status for a biallelic variants in the GCI, although I can pause for a second. Does anyone have any questions related to that? This has been available for a little bit now, the ClinVar entry. I should say it also displays very nicely in the website display that you'll see the ClinVar entries and the granular evidence that we have. Okay, so we'll move on for entering phase status for biallelic variants. So for autosomal recessive or semi-dominant gene curations, you enter biallelic variants at the case level, and you have to answer additional questions about the variant's phase because there are those two variants. So move these floating variables. Let's see. Okay. So for score, this phase status is for scoring probands for autosomal recessive conditions. It's now required. And you will have to check a box. And if, of course, biallelic cases for semi-dominant. So the curator has to check a box to attest that the variants are either homozygous or proven or suspected in trans. And so this is a screenshot here of that. So check here if there are two variants that are proven or suspected to be in trans with respect to one another. And if you wish to document information on variants that are not in trans, please add details to the non-scorable evidence section in the PubMed ID summary. So there's no option for variants in cis because two variants in cis would not be informative for autosomal recessive curation. You would not score a case as a bona fide biallelic case in that instance, or you would have to check if they are homozygous. Once the curator confirms that the two variants are proven or suspected in trans, there'll be a second set of checkboxes where the curator has to choose if the variants are proven in trans, suspected in trans, or phase unknown. And that is displayed here. If a curator selects phase proven in trans or suspected in trans, the variants must be provided to be able to score that evidence. Otherwise, you'll have a following error that's shown two variants in trans, please provide two variants, and you won't be able to save. So if you've checked this by accident, uncheck it and then check out homozygous if you only have one variant. And once two variants in trans are entered and scored, the record can be saved. And so you'll see here the two variants. And just to give a note, the phase is for a maximum of two variants at this time. We can't list the phasing of more variants. You can add the additional variants and free text about the additional variants and additional information about the individual if you wanted to provide any information about multiple variants. Because I know in a lot of publications, they'll say this individual had three variants in this gene. However, we think these two in this phase are the causative ones. So you can still include that in the notes, but we just don't support adding more than two variants and listing their phase granularly. If phase unknown is selected, you can still enter the variants observed and save them, but there will be no option to score. And really, we do suggest that you have a high suspicion of variants being in trans or they are confirmed in trans to be able to score to really give that gene disease validity evidence, especially for an autosomal recessive gene disease relationship or semi-dominant if you're going to count the biallelic case. So that's why we've kind of put this block in. If you really are entirely unsure of the phase, you probably shouldn't be scoring those variants in the first place. Because the variants aren't scored, they will not appear in the preview evidence scored tab or in the classification summaries scoring matrix. If you want to document these in a way that they will display on the website, if you really truly don't know the phase of the variants, we encourage you to enter this in the non-scorable evidence section for our PubMed ID. That way, this will actually display in the non-scorable evidence section within the table that shows up on the website. And here's the phase status viewable in the preview evidence summary here, proven in trans, for example, for these two. And now we have nice bold biallelic compound heterozygous, which is also nice. And for the second individual suspected in trans, also nice bold biallelic compound heterozygous. So that is phase, and that of course will also show up on the website. This is just an example of like from the GCI, but this will show up on the website too. Makes it a little bit easier to look at cases with multiple variants and figure out the phase. Any questions about that? Okay, then let's move on to this very convenient feature that this need has really come up often recently is editing published evidence summaries alone in the GCI. So sometimes it becomes necessary to make small edits to a summary after publishing. Might be a variety of reasons for this. Typographical errors, if a curator had entered the date incorrectly. I think what happens a lot is a GCEP would like to be added as a secondary contributor or approver. They might need to add a little bit of additional context from their disease area to your evidence summary. So this may come up. And so these types of edits can be made using the editing strategy that has already existed previously. However, if you have a curation that's done prior to SOP 8, this would prompt you to rescore the entire curation using the current SOP, which it brings a bit of administrative burden there and curation burden. So in late 2024, we released a feature that would allow these small edits that we were listing above, so typos, erroneous dates, adding secondary contributors to the evidence summary without the need for rescoring the entire curation. And for these types of changes, because we designated them as minor changes, they'll result in a minor incremental version change to the curation record when they're published to the website. That being said, if you need to make major changes, you really need to go back and change the curation, update the curation, and that would be a major version change. It's really for the things listed on this slide. So to edit a published evidence summary, first you would click View Classification Summary button in the GCI, the arrow here. And then you would click the Edit Published Summary button here. Clicking this button lets you edit the published summary, and this disclaimer alerts that only administrative edits can be made. You should not be changing the scoring. You should not be trying to override the classification, any of that. And then this very nice link here, and we'll show it in a second, takes the user to a new tab that lists all changes made to the published summary. So basically, kind of like a type edited version, so you can compare side by side. So you would make your selected edits in the Edit Published Summary section panel that opens after clicking the button. The fields that can be edited are in white here. So approval review date, secondary contributors, secondary approvers, and evidence summary. And you have to click on the pencil to edit the evidence summary. Fields that cannot be edited are grayed out. So the date that it was initially published, the new published date, this auto updates upon saving, the curation reason, administrative updates is the only permissible curation reason for this because we said there can only be minor changes here. And the published submitter name, this auto updates upon saving, so you don't have to play with this. So once you have edited it, once you've reviewed it, you would check the box to confirm the information is correct. And then by clicking save and republish, the published record will be updated and sent to the website. However, you can view the published edits history first to make sure that everything looks okay, if that helps you compare side by side a little bit more easily, because clicking the save and republish button just closes the modal and returns the users to the prior page, and it would just be updated and in theory be updated to the website. So if you click the view published edits history link, you'll get this side by side here of like a markup essentially. Sorry, this is really fuzzy. But removed text is highlighted in red, added text is highlighted in green, and changes to other fields are not highlighted. So you would just have to compare side by side here. But this can be helpful to make sure that you've really changed all the things that you needed to change before just approving it and publishing it straight to the website again. Okay, so those are all of the quick review of the updates that we had made semi-recently. Does anyone have any questions before we go into the Q&A from the survey that we had?
Hey, Marina, are you going to go into like more advanced edits, like not easy edits. We need to change an inheritance pattern for one of our curations, and I'm trying to figure out even how to like unapprove it to maybe make changes, and like whether those changes get made in the gene tracker or the GCI. Is that something you guys have time to go over today, or?
Yeah, we can answer that, Chrissy. So in our SOP, I believe one of the like first tables we have, we indicate what mode of inheritance can be transferred and cannot be transferred. So I'll just let you know we can transfer between autosomal dominant and X-linked. We can't transfer between anything else. So if you're in a situation where you're trying to go from autosomal dominant to a recessive or to a semi-dominant, it will require you to create a new gene curation interface record. You can unpublish your old record and it can sit in your GCEP's affiliation. If you feel that it becomes too confusing, you can email the GCI help desk and they can put it into like a little parking lot area to move it out of your affiliation. As far as the gene tracker goes, there can be one of two options there because we have each gene tracker record that is linked to a GCI, so we can support publishing that pre-curation information linked to the final gene disease validity classification. You can either create a new record and you can have those GCI links created and that pre-curation record as you add in the gene, disease, and mode of inheritance and move forward with your new record. If, however, you wanted to keep the record in pre-curations, we can unlink the GCI record that you had, and we can be able to create the new link and update your mode of inheritance. So there are two optional ways there, but at least for the gene curation interface, we can't transfer it. What we could suggest is that for groups, it is re-entering information, but you can download the evidence summary, and you can kind of help copy and paste the information that you have across that. It's just a really huge developmental lift to try to fill those spaces, especially between certain mode of inheritance.
Okay, yeah, unfortunately, we got to go from dominant to semi-dominant, so I guess we'll have a big overhaul. Okay,
Yeah.
well, thank you for going through that. We may be reaching out for some help. Thanks.
Yes, great. I'm happy to help and really great question.
Okay, so let's move on to our first question for scoring variant level evidence. And this is a good question. It does come up often. So this is related to scoring functional evidence. So for autosomal recessive disease, we may include the same variant more than once. For example, count a homozygote and multiple compound hets with a different second variant, but that same original variant. If there is functional evidence for the variant, let's say we would count the variant for 0.5 points. Can we count each instance of the variant at 0.5, or is that double counting the functional evidence? I.e., is it safer to count subsequent instances of the variant at 0.1? And so I think this question has a couple things in here that I'd like to unpack, but it's a very good question because I think it does come up in many GCEPs. So in general, regardless of the mode of inheritance of your curation, a functional assay can be used to upgrade a variant each time that variant is observed, because it's supporting the suspiciousness or likelihood of alteration of gene function of the variant. And oftentimes, assays can be expensive. If something is already published once, a researcher doesn't necessarily think they need to repeat a functional assay to prove that a variant is suspicious. And so we feel that this could be used each time to upgrade the variant because it is evidence towards the variant's suspiciousness. However, you really should exercise caution when scoring recurrent variants because these may be indicative of another issue. It might be indicative of a founder variant in that case, especially if you're seeing the same variant over and over and over again. And you don't want to overcount that. At a certain point, GSEPs will often discuss and kind of cap how much they would score a particular founder variant, particularly if it's in a certain population over and over again. Also, if you have the same variant in homozygosity a bunch of times, you want to make sure it's not the same patient reported multiple times as best you can. Oftentimes it's nebulous, and if it's unclear, we tend to err in the side of caution and undercount, especially if it seems like the institutions are from the same country or same location in the US, or if there are overlapping authors and you have multiple homozygous observations with just not enough clarity if it's a different person. Scoring a recurrent variant is sometimes less concerning if the variant's observed de novo multiple times, and I think these can all be scored. It's also less concerning if there are multiple compound heterozygous observations with a variant in trans with different suspicious variants. However, in general, when possible, as many unique variants should be scored in a curation as possible, although we know that in some curations or of really rare conditions, this can be a limiting factor. So we recognize that you might have to score recurrent variants, just use caution.
Yeah, I would just add to Marina's, it is really important, regardless of, the inheritance pattern, that if you're seeing these recurring variants, to make sure it isn't the same person. And often in some of your specific clinical domains, you may have lots of case studies that have been repeated. I know this is an issue in the cardiovascular section. And so really, as biocurators, make sure you're in those materials and methods and looking to see where are they getting those case cohorts from, if that's one of those situations, because it could be the same basis of that cohort, and you could potentially be scoring the same person over again. So definitely an important area to look in the paper.
I just had a quick question that I wanted to follow up on from that idea of scoring variants where it's the same variant over and over again associated with the disease. I know this comes up quite a bit in some GCSEPs, particularly for disorders that have a gain of function mechanism where it might only be like a residue or 2 or 3 variants within that gene that can cause that gain of function effect and therefore cause the disease. So is there guidance coming around, so that's a different situation. So is there specific guidance coming for how to address them? And does that even count as a gene disease relationship when really it's a relationship of a variant to the disease? Sorry for the tricky question.
Oh, no, that's very good question. And of course, as you said, Alicia, there's a number of groups that have dealt with this where it might be one variant, right? Or even one amino acid that has multiple changes, right? A couple of different genetic variants, but the same amino acid. It is really important, as Marina has emphasized several times, is you check with your GCSEP. You all can end up scoring those at the same score. What is really important, though, when it's the case of a single variant or a single amino acid, that your evidence summary makes it exceptionally clear that you only see it with those variants or that amino acid change. Because what you want to be careful of is if it does get up into that strong and definitive, that does mean, right, that this variant's going to be looked at on testing panels. It's going to have results returned. And what you don't want, right, is a bunch of pathogenic variants that aren't this variant. So I think those are where the GCSEPs have done a really good job in that summary of indicating, right? It's only this particular variant. So if you think that's something happening, alert your GCSEP of it and have that discussion.
Thank you.
Yeah, I think in a lot of those disease areas, or some of those disease areas, you get lucky, right? Like achondroplasia, where like almost many of the observations are de novo. So it's just even more evidence that that's a very suspicious variant. But you're not always lucky with that, and you just might have a bunch of inherited observations of the same variant. Okay, to continue the really good questions we got. So now we're going to talk about consanguinity and scoring consanguineous cases. So the question is, I have seen curators downgrade points for autosomal recessive disorders if parents are related in case the phenotype is caused by another recessive disorder. Is this necessary when the proband has been analyzed by exome sequencing and where presumably most other causes would have been ruled out? So I'd like to pull apart a few bits of this question too, because I have seen GCSEPs that downgrade for multiple pieces here. So that they have a standard downgrade for consanguinity, and then they have a standard downgrade for homozygosity, and that they'll pile them on top of each other. I would argue that you should only do one downgrade. So in general, there is rationale for downgrading for consanguinity, regardless of the variant type or sequencing methodology, as even exome sequencing or genome sequencing can miss variants. For example, I only sign out genomes in my clinical work. We only go 15 base pairs into the intron. So really, we're not using the full capabilities of genome sequencing there. So things can still be missed regardless of the technology. Obviously, it's a lot better than just like a single gene test, but still. The reason that we suggest this downgrade is because there are large stretches of homozygosity in these cases, often making it difficult to distinguish the true cause of disease if multiple homozygous variants are observed across multiple disease genes. Additionally, this might potentially preclude the authors from looking for de novo variants. I have had consanguineous families that have de novo variants that are a cause of their disease. So these might not be looked for in these cases too. So that might be another thing to flag as a reason to downgrade in a consanguineous situation. So I am coming from the hearing loss GCEP as one of the older GCEPs, which really does cover a disease area that has a high percentage of genes with consanguineous cases. So at least in the beginning here, we deducted a standard point amount for consanguinity alone, which is half a point, regardless of the variant type, and we don't penalize a set of variants further on top of that. So we do pose that for your consideration in other GCEPs. However, this is not required, and you should always consult with your GCEP and make sure to make a consistent scoring decision across your gene curations. Don't downgrade in one and not downgrade in the other, or downgrade really severely one time and not in the other. You really should try to be consistent, especially if you do have a disease area that has a lot of relatedness or consanguinity in that particular area.
Yeah, and I would just add one of the things that we produce out from the Gene Curation Working Group is that GCEP protocol now, and one of the items in there is developing as part of your GCEP a scoring recommendations guideline. So if you know that you have several genes and these are the questions to help with consistency, please discuss with your GCEP developing those scoring guidelines. It can help every time you have a curation to look, okay, when do I need to score down? How much do I need to present? And then that helps you all to keep track. I know in hearing loss, you all had hundreds of genes, other of your groups may have 10 to 20, but regardless of that length, that can be really a helpful process. So we can plug in the GCEP protocol link again, but just know it's there, and there is a folder that allows you to see not only a template, but some examples from other gene curation expert panels.
Yes, that's a great point. We definitely suggest to take advantage of other work that other GCEPs have done and their SOPs, especially if the disease area is similar. So I think it's over to you, Courtney. So I'm going to stop sharing and let you share for the rest of it.
Great, thank you. I will get myself together. All right, let me know. Can you all see that?
Yes.
All right. So I'll be taking over for the rest of this talk, and we'll go to our next question, which was about scoring experimental evidence, and a question really, I think, that came between how do you score biochemical function, and it brought in protein interaction as one of those potential options. So our question was for biochemical function A, where we state a gene product has the same function as the product of another gene that has evidence of involvement in the disorder. Could or should we be counting genes encoding proteins in the same biochemical pathway? So the example here being they don't have the same function, but are involved in the same overall process. So really great question. You all as biocurators are faced with a lot of information, and you're trying to figure out what is applicable and what is not. And so in this case, I think the answer would be that the genes in questions appear to be interacting in a cellular pathway, which indicates a consideration of scoring underneath protein interaction. So I'm going to go through after this a little bit more about biochemical function A and B, and then that protein interaction and some resources, hopefully making that helpful. So again, you have two ways of scoring biochemical function. A, as we said, is shared with other known genes and the disease of interest. In this situation, we really want you all to make sure that the other genes with a similar function also cause the same phenotype, and that these can be scored as relevant data. The SOP also says that that basically requires a curation of that other gene to meet a strong or definitive classification. So if that is the situation, you may be having to help your gene curation expert panel, or if it's for another panel, look and see if those curations have been formed. Some that work really well within this is when we think of ion channels, right? Those are genes with similar functions, and those can usually result in a similar phenotype, scaffolding proteins, etc. The second biochemical function, B, is that you have a function consistent with the phenotype. So an example here may be if your phenotype you observed is cancer and the biological function of your gene of interest is a tumor suppressor, then it is logical that variation within that gene can alter the tumor suppressor function, thus causing cancer. So it's one of those kind of logics about what the function is of that protein product and how that would play a role in the development of that disease. So again, making sure that for either one of these, the function of the other gene must be that definitive gene disease classification, and that's mostly in A, right? B is really talking about what is the function of that gene and its protein product in relation to the mechanism of that phenotype. Now, how are some ways that you can potentially find out genes, right, that would fit in biochemical function A? Well, let's think about some resources you're already using. So OMA may be a way and be really helpful for you to assess your gene of interest as part of a phenotypic series. So if you've not ever seen this before, if you go to your gene of interest and you look at that phenotype and click that phenotype min number, you may find a little button that says blue phenotypic series. If you click that, this is what you're going to see, and it will show you a bunch of other genes that have been asserted to be associated with a phenotype similar to what you're looking at. So what you can see in the orange boxes here are that I had an interest in long QT syndrome from potassium channels. And what I can see is there are several potassium channels that are already indicative of long QT syndrome. So again, this is one of those areas where you potentially can use a resource you're already in to think about other genes. And then check that they're on your gene list and how they've been curated, and then work with your group whether you need to have additional curation to help apply that biochemical function A. Another place that you can find this, in biochemical function A, you do have to enter in a gene ontology ID. And so that Go term is another place you could go to. I know sounds really funny. So Go has several different ways you can look up the ontologies to see what that specific function is. So there's Amigo, which allows for a search by a gene, and then the Go term, and there's also Quick Go. What you're looking at here is Amigo. I put in a Go term for specific ion channel, and I looked at its function. It is to involved in ventricular cardiac muscle. After I did that, I could look at a list of the genes that also came up with that same Go term. So since you already have to find a Go, if you're looking at what are the other genes, that might be helpful for you to see what genes could be involved in the same type of phenotype and the same type of biochemical function. Now, getting back to the question, we've got a whole cellular pathway. So again, evidence showing a gene product interacts with proteins that have previously implicated in the disease of interest, that EP can consider if the protein is part of a whole entire signaling pathway that's associated with that disease and score it within protein interaction. Again, many times genes can be co-immunoprecipitated as part of a complex, which indicates an interaction, even if it's not a direct interaction, right? We do write that in the SOP. So again, signaling pathways regulating cell division. You're seeing an example on the right here of cancer genes and how we can look at DNA synthesis and repair. And again, that there are several proteins along this pathway that may have different functions along the pathway, but together in the signaling pathway result in cancer. Again, we also see mismatch repair, MMR, as a really great example. So these are places that you can look to score that. And of course, make sure you have a PMID, and that PMID can still be a review that goes over that cell signaling pathway. Another resource, though, that you can think about as looking at cellular pathways and what other proteins might be in that signaling pathway within a cell is this StringDB. We'll put this out, and afterwards we'll consider updating this in the SOP, but it's a really cool tool. If you go to stringdb.org and you search by your gene, you'll be presented back with this really cool thing. Figure you can see different proteins, but what I would guide you to do is go to the analysis tab. From analysis, you're going to see those Go terms. So another place you can get the Go ontology, that's really important, and you can search by that. But if you keep scrolling down a little bit more, you'll get to this PMID and reference publications. So great. Now you may have publications to use to be able to score this evidence. So another resource of pre-curated data that could help you to expedite those curations and get that experimental evidence. Any questions on this before I move to our last question of the topic we had and open it up for other questions? Great. So our last question was really addressing with the fact that we have more AI and large language models, what are some things that ClinGen may be thinking about for gene curation? And so I want to talk a little bit about that today and get some feedback. So right now, ClinGen is in an exploratory mode for the uses of artificial intelligence, machine learning, automation, anything that's a computational method for ClinGen curations. But we want to note that this technology is moving so fast, this will be true for some time. So we want to be very thoughtful about where we want to put our time and energy to help promote the use of this to help you all out, and that we're not changing things over and over and getting you all confused as users. So we also recognize some groups within ClinGen will be working on AI-based tools to support curation efforts. In fact, we have several different efforts between looking at literature searches at multiple different areas that you could use, again, to expedite finding that literature and prioritizing to be able to move forward with curations. We also understand that some of you may already be using AI and machine learning based tools outside of this system to help with your curations. So we're going to be really interested to know from you all what you are using, what you like, what you don't like. And so we are going to send out potentially other surveys to the Biocurator work group in the future just to be able to hear. Certainly you can share some today. Again, we're trialing some of our own tools, and if all goes well, we'll share them with you all as a wider group of curators. Part of that is because we have formed this new AI/ML subgroup to coordinate activities. Now, the one thing I do want to tell you about with AI and ML, because it is moving so rapidly, we know there are multiple different resources you can get this information. But we also know the fact that because of its training sets and other things, it can hallucinate. So we do want to indicate if you are using AI or ML for your curations, we really want to have you all have caution in using that and where you're using it. For example, if you're using it to generate your evidence summaries that you're presenting to your expert panels, please, please double check and read those over. Make sure that those references that could be included are accurate. The last thing we want is to go ahead and move through with publishing evidence summaries and they have inaccurate information there. So that is something for you all to just understand as you're using this word of caution and make sure you're going back and validating your information that you're receiving from these resources. But again, we may be interested in assessing whether or not what you're using for AI and having trials. We also would like to run monitored trial of the technology before applying it in curation. So in the meantime, most DSEPs for you all have developed, in my example, the evidence summary. You've already generated that text. There's a standard evidence summary text, so please utilize that in the process. But if you do have questions and interests, we certainly want you all to contact us. So that's going to be my next slide. If you want to participate in any experiences with AI, if you want to be able to let us know how you're using it, how it's been helpful, what concerns you may have, you can email us at genecuration@clinicalgenome.org. And again, this AI/ML subgroup of ClinGen is working to draft a survey in the weeks to come to ask you about how you all are using it. And I think that's the end of our questions that we received so far. So happy to open it up for questions you may have about gene curation, or if you all want to share any of your thoughts of how you are using AI or ML for gene curations at the moment.
Could I bother you with a quick question, Courtney, about, like, I noticed you had a slide on protein interaction evidence.
Mhm.
Can I bug you with a question that might be like double counting related?
Sure.
We were curious, like one of the groups told us that for the biochem A category, when two proteins are like maybe in the same complex and you've curated one of them already, it's definitive. They said sometimes they like to reward that protein-protein interaction with like biochem A, but they were curious if that would be like double counting to then reward it with protein interaction as well. Do you have like a strong opinion about that?
Okay, so good question of what I'm hearing. I think it depends on the situation. So if the proteins that are interacting with each other can be protein interaction, also have the same biochemical function, then you could count the biochemical function as well. So this could be things where potentially you have two receptors, right? And those receptors have to dimerize, and that dimerization leads to the same cell signaling. They've interacted, and they're both receptors that have to function in the pathway for disease. You could even, if the question is that the information's coming from the same paper, then yes, use the same paper, right? And extract the information there. I don't think that that's necessarily double counting. Where it would be a problem is if you're saying because they interact, protein interaction score, yes, but now I'm saying they have the same function, might not be accurate, right? So you could have a receptor that's also interacting with the scaffolding protein. They have two separate functions. You would not do biochemical function A there. You would only do the protein interaction. Does that answer your question, Bill?
That makes sense. So basically, there's scenarios where there's basically two pieces of information there, and you're rewarding them separately. That sounds great. Thank you.
Continuing on that theme of protein interaction related questions, and apologies if you covered this, but firstly, the idea that the gene already has to have been curated as strong or definitive, does that have to come from the ClinGen group as having curated it, or can we be using resources like GenCC? Like if another reputable source has curated it, are we happy to use that?
I don't think we have any hard and fast rules. So if it has been from a reputable source like GenCC and you as a GCEP feel comfortable with the information that is there, you think you could move forward. I don't think there's anything in the GCI that, categorizes this as strong or definitive and you move forward. However, that's one of those cautionary points. Just make sure that you're looking at the data that they have selected. You agree, right, with that classification. It might be something too where with GenCC, are they all aligned? Everyone who's entered the information is all aligned on that classification. If there are ones that are conflicting, that might be something you want to have caution for a group.
Yeah, I agree. I think I definitely have GCEPs who use GenCC, especially if they're just in the beginning stages of their curation, that they don't have a ton of genes curated. But as like a beginner primer to GenCC, you would either want to use concordance like this, or often I will suggest clinical testing labs because they have the most similar curation framework to ClinGen. And so if there's a discrepant, research organization within GenCC, that might not be as concerning. But if the clinical labs are discrepant, I would say take that with a big caution there.
And a second question around protein interaction for the idea that the default score is half a point, but then there's obviously the option to upgrade it all the way up to 2 points. So is there like sort of a standard approach to what is required to upgrade? Like, does it need to be seen with multiple genes in that same pathway and like lots of genes qualify for an upgrade, or is it like these are the only two genes that interact? And but like how is there sort of guidance to how to qualify when an upgrade is warranted?
Yeah, that's really good. And I think it shows the flexibility of the framework itself and just again, the flexibility we allow you all as GCEPs. If it is only two proteins, right, you're not going to get multiple other proteins to help. But what you may want to look at there is, has it been confirmed by more than one institution, authors, or papers, right? So if you're getting claims that has happened multiple times and they're consistent, you can consider scoring up, right? I think you can also do that when you think about, yep, there are multiple in this pathway, you can add those to score up well, just to, again, solidify the fact you're seeing the same process. I think you could go either way with that. Marina, do you have any other thoughts about upgrades?
No, I think that makes sense. I think the most common one is like multiple genes is when I've seen the upgrade.
Are there any other questions related to gene curation from people on the call? There's not anything that I could see in the chat, but just checking back through that I haven't missed something.
Definitely feel free if you all, with these new technologies and new functionalities we put out, if there are any issues, you can always email ClinGen or Gene Curation, right, at clinicalgenome.org. Please make sure you're emailing the Stanford Help Desk. Those come back to us and we are able to help resolve any of those types of issues that you have. And of course, please continue to work with your GCIPs and update if there are things you think would be helpful. Marina and I participate in other groups with gene curation with several of you here, and we're always wanting to make sure that we are making appropriate updates to our systems to help you all with your curations.
Wonderful. Well, if there's not any other questions, then we might wrap up early and people can have a few minutes back to their day. So thank you again so much, Courtney and Marina, and we will meet again at the end of the month for an update from, I have now forgotten who it's from.
The DAPC.
That DAPC group, working group. So we will see you at the end of the month.
Thanks all.
Thanks,
Bye.
everyone.
Okay.